The ketogenic diet has gained significant attention not only for its effectiveness in weight loss and metabolic control, but also for its potential to enhance cellular health through mechanisms such as autophagy. This high-fat, very low-carbohydrate dietary approach shifts the body's primary fuel source from glucose to fat-derived ketone bodies, a metabolic state known as ketosis. During this transition, several powerful cellular pathways are activated, creating a biological environment that promotes cellular recycling and renewal. When carbohydrates are severely restricted, blood glucose and insulin levels fall dramatically. This drop in insulin mimics a fasting-like state, which in turn leads to the suppression of the mTOR pathway, a major inhibitor of autophagy. At the same time, the energy stress induced by low glucose availability activates AMP-activated protein kinase, AMPK, a master regulator of cellular energy homeostasis. AMPK stimulates autophagy by phosphorylating downstream targets that initiate the formation of autophagosomes, the structures responsible for engulfing and degrading cellular waste. In parallel, the ketogenic state activates sirtuin, 1, SIRT1, a NAD-dependent deacetylase that regulates a broad range of metabolic and stress response genes. SIRT1 plays a key role in promoting autophagy by deacetylating autophagy-related proteins and modulating mitochondrial function. It also supports longevity-related pathways by reducing oxidative stress and inflammation. The dual activation of AMPK and SIRT1 represents a powerful biochemical signal for autophagy induction, making ketosis an effective metabolic condition for initiating cellular cleanup. One of the major benefits of the ketogenic diet is its ability to provide a continuous low-glucose environment while maintaining adequate caloric intake, which can be more sustainable than long-term caloric restriction or fasting for many individuals. In animal models and emerging human studies, ketogenic diets have been shown to protect against neurodegenerative diseases like Alzheimer's and Parkinson's, reduce seizure frequency in epilepsy, and improve mitochondrial function, all outcomes that are partially mediated through enhanced autophagy and mitophagy. Nutritionally, the ketogenic diet can be optimized to further support autophagy by including specific fats that promote ketone production and cellular resilience. Medium-chain triglycerides, MCTs, found abundantly in coconut oil and available in concentrated supplemental forms, are rapidly converted to ketones in the liver and provide an efficient fuel source for both body and brain. Unlike long-chain fats, MCTs bypass normal fat digestion and are absorbed directly into the portal vein, stimulating ketosis even in the presence of slightly higher protein or carbohydrate intake. This makes them especially useful for individuals transitioning into a ketogenic state or aiming to maintain deep ketosis without excessive dietary rigidity. Another key supportive nutrient group includes omega-3 fatty acids, particularly EPA and DHA, which are well known for their anti-inflammatory effects and their role in maintaining cellular membrane integrity. These fats have been shown to synergize with a ketogenic diet by enhancing mitochondrial function, reducing oxidative damage, and promoting neurogenesis. Omega-3s also influence autophagy through several molecular channels, including PARs and FOXO-transcription factors, which regulate genes involved in cellular stress responses and longevity. Coconut oil itself is rich in natural MCTs and also contains lauric acid, which exhibits antimicrobial properties that can help support gut health during dietary transitions. Including sources of monounsaturated fats like olive oil and antioxidant-rich plant-based foods such as leafy greens and herbs can further buffer the oxidative load and ensure micronutrient adequacy, which is essential for safe and sustained autophagy. It is also worth noting that ketogenic diets can increase levels of beta-hydroxybutyrate, BHB, one of the primary ketone bodies, which functions not only as a fuel but also as a signaling molecule. BHB has been shown to inhibit class I histone deacetylases, DACs, leading to increased expression of genes involved in stress resistance, detoxification, and cellular repair, many of which overlap with autophagy-related pathways. This epigenetic effect adds another layer of benefit to the ketogenic state, particularly in tissues like the brain, where BHB can cross the blood-brain barrier and promote neuroprotection. In conclusion, the ketogenic diet represents a metabolically strategic approach to triggering autophagy without the need for outright caloric deprivation. By down-regulating insulin and mTOR while upregulating EMPK and SIRT1, Ketosis promotes a cellular environment of repair, renewal, and resilience. When combined with key fats like MCTs, coconut oil, and omega-3s, this dietary pattern supports not only metabolic health, but also the intracellular processes that contribute to longevity, disease prevention, and cognitive function. As our understanding of nutrient-gene interactions deepens, the ketogenic diet stands out as a powerful tool for harnessing the body's intrinsic self-cleaning systems.